Dear friends, let's have a brief presentation on cytokinin signaling. Cytokinin, a type of growth promoter in plants. Now this is the core mechanism which explains how cytokinin mode of action can be regulated. The whole signaling is known as two-component pathway, as it is mediated by two prominent proteins, namely histidine kinase receptor HK, response regulator RR. Let us discuss every point in the terminology in a detailed manner. Now first of all, let us define what is ligand. Ligand here used is cytokinin. It initiates the pathway and acts as signaling molecule. Now what is receptor? The receptor is a protein which receives the ligand. Here the receptor is CRE1, means cytokinin response 1 protein. However, in Arabidopsis, receptor has different types. The receptor used is AHKS, means Arabidopsis hybrid His kinase. It has three important forms: AHK, 2AHK, 3 and AHK4. Receptor molecule Arabidopsis hybrid His kinase. This is also known as sensory histidine kinase. It plays significant role in the whole mechanism. The receptor, as the word indicates, combines with the ligand to form ligand-receptor complex. The whole complex gets activated by means of ATP through the process of autophosphorylation. My dear friends, as we have already discussed, sensory histidine kinase is a complex type of protein. So in order to understand the topic beautifully, let us go for a complete structure of sensory histidine hybrid kinase. Its diagram is like this. It has outermost region known as J's domain, found towards extracellular part. Here the ligand binds. Now one more part is called His kinase domain. It has histidine residue, it gets autophosphorylated and gets activated. Then last part is called as receiver domain. Receiver domain has aspartate residue, it gets phosphate from the His kinase domain, hence whole complex gets activated. Now one more important protein used in the whole signaling is known as AHP, Arabidopsis histidine phosphotransfer protein. This protein has well-defined structure. We can say AHP. It also gets phosphorylated. It acts as a mediator protein or a middleman. AHP protein gets the phosphate group from the receiver domain and passes to the nucleus. Hence we can say it acts as an intermediate messenger. Now proceeding with the last part of terminology, this is Arabidopsis response regulator ARR. It has two important types. Number one, type B ARR and type A ARR. Now what is the difference between two proteins? Let me show you first of all diagrammatic representation. This is the diagram of type BARR. It has two important regions, DNA binding region and receiver domain. The receiver domain has well defined what we call it as aspartate residue, while as type A protein has only receiver domain, it lacks DNA binding region. Type B protein is longer, it acts as a transcriptional factor, while as type A is short, it acts as a repressor unit in the whole pathway. So we can write type B has positive control on the signal, while as type A has negative control on the signal, which indicates type B helps activation of cytokinin responsive genes. Type A dampens cytokinin response, it halts the process. Now in the last part of story, let me tell you what happens in the mechanism, how the whole cytokinin signaling is driven up. Let me show you diagrammatic representation. This is cell membrane. On this cell membrane, we have two monomers of CRE1 or AHK receptor. Each receptor has J's domain towards extracellular region, while as His kinase and receiver domain towards the intercellular side. Now towards the inner side, there is well-defined nucleus, which have what we call it as DNA molecule. Now the DNA molecule has a response regulator, namely type B ARR protein, which acts as a transcriptional unit. Now, initiation of signaling takes place when the ligand in the form of cytokinin binds to the chase domain, which leads to dimerization of receptor molecule. This dimerization leads to activation of the His kinase domain of the receptor. Then the activated His kinase acts on ATP and gets phosphate from it. The phosphate group of His kinase gets transferred to the aspartate residue on receiver domain, like shown in the diagram. Now there is one more protein, AHP protein. It is inactive form. It gets phosphorylated by receiver domain of receptor. AHP receives the phosphate by means of histidine domain. As AHP is the mediator, it translocates from cytoplasm into the nucleus. Now here AHP passes phosphate to the aspartate residue present at the receiver domain of type BARR. Now type BARR gets activated and leads to transcription. The transcription of DNA leads to formation of mRNA, or we can say messenger RNA. Later on, messenger RNA changes into proteins. And a very important protein formed in this pathway is known as type ARR. Some other types of genes are also gets expressed, for example, cytokinin responsive genes. These cytokinin responsive genes perform different functions in the plant body. For example, they help in cell division, apical dominance, also in nutrient allocation. Now at last, type ARR protein, it also gets phosphorylated by AHP and it stops the whole pathway. It stops translocation of AHP from cytoplasm to nucleus. Dear friends, it was all about cytokine signaling. I hope you got this concept by all means. Thanks, have a nice day.